skin lesions results in numerous psychological and functional problems, and their treatment creates a coverage that supports repair and restoration of skin functionality. Normally, we use mesenchymal skin cells in tissue engineering because they have a renewed capacity, multipotentiality, and the ability to release active molecules important for the tissue repair. This article shows a new strategy for tissue engineering and cell therapy. They used human skin-derived mesenchymal stromal cells that display similar characteristics of the multipotent mesenchymal stem cells. These authors developed a 3D culture system of human skin-derived mesenchymal stem cells with dermal substitute, Integra and Pelnac, in which cells adhere and proliferate while maintaining mesenchymal stem cell properties and gene expression of multi-lineage markers. This article shows a promising effective tool in vitro for tissue engineering and regenerative cell tissue technology. First, I chose this article because in one of our classes we talk about skin regeneration, and I was surprised in the development in this field, so I make a little research on the area. I found this article showing a new strategy for tissue regeneration, and I found it very interesting. The skin-derived mesenchymal stromal cells isolated from human skin demonstrate to exhibit mesenchymal stem cells characteristics such as cell phenotype, transdifferentiation potential, and immunosuppressive properties, and thus are functionally comparable to mesenchymal stem cells. After reading the article, I think that this new strategy for dermal substitute will be very useful in the future repair skin injuries, chronic ulcer, deep wounds, and other diseases. In this article, the authors show that the skin-derived cells are similar, but not the same as mesenchymal stem cells. Human skin-derived cells display characteristics as plastic adhesion, fibroblast morphology, mesodermal differentiation capacity, expression of mesenchymal markers, and absence of hematopoietic markers. How is that possible? Well, they do a lot of experiments and demonstrate that these cells are positive for mesenchymal markers, but they are negative for hematopoietic markers, as we said before. In addition, they express a neural and mesenchymal marker, but not endothelium. If they are not the same, but they can help in regeneration, could this mean that not only in the skin we have this possibility? I think that this finding is such an advance in regeneration field, but we cannot discard that this process is being done in vitro and not in vivo yet. So we have to wait for more research in the field. Another important thing that they say is that 40% of the skin-derived cells are positive for alpha SMA, marker of smooth muscular cells, also related to vasculogenesis. In their opinion, this is related to multipotency of these cells. But if only 40% express it, could this tissue also regenerate? How much percentage we need to have the capacity of regeneration? And could this cell develop and grow a cancer, given that they show characteristics of multipotency? The cells demonstrate adherence to both matrices used, and they establish also strong connections, and they maintain typical fibroblast morphology, so they don't have a problem growing the two matrices. They use Pelnac and Integra as a dermal substitute to grow the cells, but in the experiment in vitro, they observe that the cells concentrated in the upper portion of the matrices. Maybe they are right, because they say that this is because the cell seeding procedure, but I think that it's not very clear. So we need to be very sure in the process. So they need more experiments for adherence in the two matrices to be sure that this will really work. In conclusion, I think that these findings are very promising in the field of skin regeneration, but they need a lot of experiments and must be secure that this will work in vivo for treating patients. Thank you.